Hi, everyone. Today we are so honored to congratulate Dr. Greg Dussor from Texas with amazing publication in Cephalalgia of recent work, which is editor's choice for the month. And please, Dr. Dussor, tell us a little bit about place where you practice and about what brought you to this research, little bit of background. Thank you.
Sure. Thank you, Nina. This is great. I appreciate the opportunity to do this, and certainly appreciate the honor that Cephalalgia gave us by choosing this article. It's really fantastic recognition, and we all the authors appreciate it, even though I'm the only one talking today. So I'm a basic researcher, a neuroscientist by background. My primary job is I'm the department head of neuroscience at the University of Texas at Dallas, where we have a preclinical research laboratory. And I've studied pain really my entire academic research career. But headache has really been the focus of the last maybe 12, 13, 14 years or so. And so, we've spent a lot of time trying to optimize preclinical models, maybe take some of the models that existed in the field and tweak them a little bit to try to make them better, improve some feature of them. We've tried to contribute some new ways to think about modeling migraine in animals. And, I should acknowledge right off the top that none of us in the preclinical research space think that you can completely replicate the human experience of migraine in an animal model. That's not a reasonable expectation. But we do think we can model aspects of migraine, and it may take, 4 or 5 different animal models to model different features and different aspects and different mechanisms. And it may just end up being something that we have to test across multiple models when somebody identifies a new mechanism. We test it across models and we see, how replicatable it is across these models, and then we get more confident moving forward, maybe into humans with something. So this is something we've been spending a lot of time on. I think the models in the migraine field and in animals we still need more optimization of them. We still need probably more of them. And one of the things that we've tried to take backwards from humans to improve the animal models is what are the common triggers of migraine in people? And this nitric oxide donors like nitroglycerin, that's a well established trigger. And so people many years ago had taken that back into animal models and tried to create animal models where you expose the animal to nitric oxide in various ways. And that's certainly a one way to look at this, and it probably has um, mechanistic specificity to things related to nitric oxide, whatever those are. But what we also tried to do was take stress, which is among the most commonly reported triggers of migraine in people, and say, how can we use that observation from humans and bring that back into an animal model and see whether we could make some mechanistic observations about how stress might be contributing to migraine. So that's a model that we've been working with for several years now. And there was a previous paper that we published in Cephalalgia a few years ago that was the sort of baseline for the work that's in the current paper. And what we showed in the previous paper was that if you repeatedly stress animals, this is the method that we've been using. We do 3 days of stress exposure for 2 hours a day to mice, and that causes them to be hypersensitive for about 10 days or so. They recover from that hypersensitivity, and after they recover, they're sensitized to various substances, various things, various other stimuli, one of them being a nitric oxide donor. And so normally, if in an animal model, if you're going to expose the animal to a nitric oxide donor, you have to use a fairly high dose to get the animals to respond. And if you do this after you stress the animals and you let them recover, now the dose of the nitric oxide donor that you need is much lower than what you need in an otherwise normal animal. And to me, that is more interesting because that's how humans are. Not all humans will get a migraine if you give them nitroglycerin. Only people who are susceptible to migraine will, right? And so what we want is a context where we can cause animals to respond to much lower doses of nitric oxide donors than what would normally be needed. And that's actually what happens when you stress the animals and let them recover, right? So I like this model because it incorporates multiple features that we know are true in humans, that we know that stress contributes to migraine in some way that's not well understood. And we know that nitric oxide contributes to migraine in some way that's not well understood. And we're trying to put those two things together. And that's where things get messy is, stress is an incredibly complex physiological stimulus. It impacts every part of the body, not just the nervous system, not just the brain. Every cell in the body is impacted by stress. And that's also true of nitric oxide. It's freely diffuses everywhere in the body, right? And it's almost in every single cell. So those are two very messy things to try to work with. And it shouldn't be surprising that if you start trying to manipulate stress hormones in a model like this, that things are going to get confusing very quickly. And that's essentially what we saw in this current paper is that, almost anything that you manipulate in a stress circuit, any of the stress hormones that we can think about is going to change the behavior of how the animals respond to stress in some way. And that's kind of what we saw here. So I've talked a lot. I'm gonna pause for a second to just see if there's any sort of follow-up questions from what I talked about, and I can keep going if you don't have any questions.
Thank you so much. That's definitely complex situation. And I think what you describing known in clinical practice that triggers could be cumulative. I also really appreciate your review of complexity and how you approach things from a scientific side. We know in clinical practice that stress is very common trigger and things with nitric oxide can also be triggered. So thank you so much for doing this research and supporting what we see in clinical practice with hope that we untangle it together and will develop treatments because of all our working as a team. So thank you so much. How did you figure out methods for this particular research is fascinating. Can you tell us a little bit about specifics which really beautifully presented in your article? It's graphical abstract right there. It's really neat and really elegant research. Can you tell us a little bit about specifics for this particular discovery?
Yeah, thank you. You're too generous. The graphical abstract was somewhat complex to try to come up with because the findings are actually complex. So, one of the things that we had found in the previous study that was the precursor to this one was that the effects of stress on the animals, if you block the synthesis of in mice and rats, the major stress hormone is corticosterone. In humans, it's cortisol. So the hormones are very similar to each other, but they're actually not exactly the same. But the function of corticosterone in the rodent is equivalent to the function of cortisol in the human. So we talk about corticosterone in a human context. You just think about that as being cortisol because they're essentially the same thing. So what we found in the animals was that if you block the synthesis of corticosterone, which you can do with an inhibitor of the enzyme that makes corticosterone, that you don't get the effects of stress, the repeated stress exposure. That's probably not terribly surprising because you would expect that in a human that cortisol would play a major role in the response to stress, and that in an animal that corticosterone would play a major role in response to stress. So we kind of expected that we would see that. Um, if you block glucocorticoid receptors, which is what corticosterone and its relatives are, you could basically get the same effect. So if you don't have corticosterone, or if it can't engage its receptor, the effects of stress are not created in the animals. And so again, not terribly surprising. And so what we asked in the previous paper was, well, okay, so if corticosterone is so important, maybe we don't actually need to stress the animals. Maybe we can just give them corticosterone, just inject it 3 days in a row and see what happens. And so we did that experiment, and what happened was that in the females, you get a response that looks like stress. It's not exactly the same magnitude, but the sort of pattern in the time course is similar, but it didn't do anything in males. So that suggested that there is something very important about corticosterone in females, but in males, there's other things. It's not just that. There has to be other factors. So we wanted to expand on that and say, um, the most likely treatment interventional time point in humans is going to be after stress happens, right? So, it's possible that you could come up with a therapeutic that in humans they would dose themselves around the time when they know they're going to experience a stressful event. And this would be people who know that they have reliable migraines triggered by stress exposure. Maybe they dose themselves before the stressful event happens. That's possible that we could come up with a therapeutic like that. But realistically, people are going to dose themselves after the stressful event is over and when the migraine is actually happening. And so we wanted to sort of probe at some of these hormones after the stress was finished. And so what we did was we gave the inhibitor of corticosterone synthesis after the stress exposures. And the question was, once stress is over and the animal's no longer being exposed to the stress, does the synthesis of corticosterone still matter at that point? And yes, it does. So there is some continual need for corticosterone synthesis even after the stressful event is over. Um, One of the complicating things that happens in this stress hormone circuit, which is very, very well organized. The body's had many, many, many years to get this right and to figure out how to do this. So one of the things that happens when you inhibit the synthesis of corticosterone in the animals, and the same thing would happen in humans. If you inhibit the synthesis of cortisol in humans, the hypothalamic pituitary axis doesn't see any cortisol circulating in the bloodstream. And in the animals, the HP axis doesn't see any corticosterone signaling in the bloodstream because you've inhibited the production of it. And what the circuit does in response to no corticosterone is it releases more of the hormone that stimulates corticosterone to be produced. And that hormone is adrenocorticotropic hormone, or ACTH. And that comes out of the HP axis, the hypothalamic pituitary axis. That gets released from that axis. It goes to the adrenal glands. It stimulates the production of corticosterone, and then you make more corticosterone floating around, right? So there's a feedback mechanism that when you inhibit cort synthesis, that you end up getting an increase in the release of ACTH. And so one of the questions that we've been trying to ask is that when you modulate this circuit, when you inhibit the production of corticosterone and you see these behavioral effects where you don't get the same behavioral response to stress because there's no corticosterone, well, there really could be two explanations for that. The first is it's mediated by corticosterone, and that's why you don't see the effect. The other is that it's also mediated by an increase in ACTH, which is now being released by the HP axis because there's no corticosterone, right? Again, this comes back to the statement I made earlier. This is an incredibly complex system, and it's going to take a long time to try to figure out how this thing works. So then what we said was, okay, well, instead of inhibiting corticosterone, what if we just give the animals ACTH, sort of mimicking the response that the circuit would have? And that worked. That blocked the the behavior. So then we said, okay, well, if we give ACTH to animals, we know what's going to happen is that ACTH is going to cause the production of more corticosterone. And so we had to inhibit then the production of corticosterone by giving the enzyme inhibitor. You see how this is this gets more complicated with every figure of the paper. And what we found was that even if you inhibit the production of corticosterone, you still get an effect of ACTH. So there is something that not only corticosterone is doing in the context of the stress response to produce behavior. But there's something that ACTH is capable of doing independently of corticosterone. Because even if you inhibit the production of corticosterone, you still get an effect of ACTH. So we know, of course, that ACTH can be hydrolyzed into alpha melanocyte stimulating hormone. So this is alpha MSH. And so we tried that. It produced a similar effect to ACTH. And then we sort of had to guess what, because there's many receptors for ACTH and there's many receptors for MSH. And we knew that one of the receptors that would potentially be important is the melanocortin 4 receptor, MC4. And there happened to be specific activators, agonists of MC4. And so we tested that, and that produced a similar effect to ACTH and MSH. It attenuated the behavioral response. So, it's very difficult to put all of this together and say, what would we do in a human if we were going to translate this into a human? There might be many aspects of this that might either work from a therapeutic perspective or actually might make people worse. We don't know that yet. I think because these circuits are so complex, and when you change one component of the circuit, it's probable that 2, 3, 4, 5 other things in the circuit also change as a response. It's going to take a whole lot more work probing at these different parts of the stress response pathway, these hormone pathways, to try to figure out who's doing what, how are they working in parallel, which one of these things might be therapeutically targetable. Because what we want to do ultimately is prevent people from getting migraines in response to stress. But what we don't want to do is prevent all of the good things that happen as a response to stress that the body does to try to adapt to the stressor itself, right? We have all of these stress response systems because it helps the body adapt to the exposure to stress. But it's probable that in some cases, these things don't work perfectly, especially when you're exposed to stress repeatedly over long periods of time. Those response mechanisms might start doing things that are counterproductive. And what we have to figure out is what are the counterproductive things that stress exposure is doing, and target those, and not target the helpful things that these circuits do in response to stress, because that's not something that we want to target. So um, this is an incredibly complex thing. We're not going to figure out in 1 or 2 papers. It's going to take a whole lot more work to do this. And I think there's a reason why we don't know a whole lot about how all of these circuits work. And of course, acknowledging that there are entire fields of research that are focused on how stress exposure and how these circuits impact different systems and functions of the body. Those fields of research have been happening for decades now, and there's still a whole lot that we don't know. I think there's a tendency, and maybe this is why there hasn't been that much work done on stress in the migraine field. There's a tendency to look at this, look at stress and say, that's amazingly complicated. I'm going to work on something else because this is a complete mess. How are we ever going to figure this out? And, I don't think that's fair, because stress is such an important contributor to migraine in ways that we don't understand. If we all say, look, stress is too complicated, we're not going to work on this, we're going to work on something maybe that's easier to focus on and be specific about, we're never going to figure out how stress contributes to migraine. So I think, yes, this paper, the findings are very complicated. It's hard even for me to explain how they work because I don't understand this yet. It's going to take more time and more work for us to do. But I think that we need to embrace that complexity and say, look, it's okay if it's complicated. It's okay that if we do a single study that we don't figure everything out. It's okay that if the results of the paper maybe don't make perfect sense, that's fine. We have to do that, because if we don't, we're never going to figure out how stress actually contributes to migraine. And so I'm hoping that, in addition to the sort of mechanistic insights that we're trying to make, that we encourage other people, either in the migraine field already or maybe in other fields, to look at this and say, okay, yeah, we should do this, right? It's okay to publish a paper where not all of the findings make sense, where it's very complicated and we can't explain everything, that's okay. We need to have papers like that because they are going to build on each other and eventually we'll figure this out. And if we don't even try, we're never going to get anywhere.
Thank you so much. I completely agree. It also supports us as in clinics when we see effects of stress or role of pituitary, role of other hormones in clinical picture. So we very grateful for your work and we looking forward for more articles in Cephalalgia and future research with you. Thank you so much. And again, congratulations with Editor's Choice Award. Thank you.
Thank you. Thank you for the honor and thank you for the time doing this discussion. It's great talking to you.